Imagine you're out in your yard, tending your garden and enjoying a beautiful day, when you realize over there in the corner is a small patch of poison ivy. Now, no one likes poison ivy. It has the potential to cause us a lot of harm and pain and suffering. You have to act, and you have three choices. One, you could take a flamethrower and destroy every living thing anywhere near that poison ivy. Two, you could rip out and kill all of the poison ivy plants you can find. Or three, you could recognize that there actually isn't anything bad about the poison ivy plant itself. And instead you could just try and figure out a way to stop it from making the toxic oil that causes the allergic reactions in our skin. Now, all of these approaches would prevent the poison ivy from giving us an itchy rash, but some are clearly more subtle than others. Now, the flamethrower approach may sound ridiculous, but it's analogous to how we use antibiotics today. We often use very high doses of what are called broad-spectrum antibiotics, meaning that they can kill many different species of bacteria, not just the specific pathogen that's causing us harm. This so-called scorched earth approach does work. Antibiotics have been tremendously beneficial to our public health over the last century. But in the same way that a flamethrower would harm the plants that you love in your yard while trying to eradicate the poison ivy, antibiotic use can negatively impact the beneficial bacteria living in and on our bodies. Even more importantly, the overuse of these powerful killing agents greatly contributes to antibiotic resistance, a massive public health problem threatening us today. So I'm going to tell you about how my lab is confronting antibiotic drug resistance by using the third most nuanced approach I described. Not trying to kill or remove the bad bacteria or pathogens, but instead simply trying to change their behavior or activity, or what we call their phenotypes, so that the pathogenic bacteria can no longer cause us harm. If we can change a pathogen's phenotype or their behavior, we might not even need to use antibiotics at all. This would reduce the emergence of drug resistance while still keeping people safe and leaving our beneficial bacteria intact. This approach is promising because we know that people can coexist with pathogens that aren't expressing their harmful phenotypes. It turns out that large numbers of us right now are colonized by pathogenic bacteria that aren't causing us any harm. So one example of one of the dangerous behaviors that pathogens adopt that can cause serious health problems is the formation of what we call a biofilm. What is a biofilm? Well, you can think of a biofilm like a bacterial city. It's a community of cells living together in close proximity, all stuck together in a sticky matrix that holds the cells to each other and attaches them to surfaces. Now, whether you know it or not, you've seen biofilms. The plaque on your teeth is a biofilm. So is the nasty slimy stuff that builds up in an unclean shower. And biofilms are very difficult to remove from surfaces because of that sticky matrix that the bacteria make. So when pathogenic bacteria form biofilms in places that they shouldn't, like on medical catheters or on some other parts of our bodies, they can cause serious health problems. Now, much like people in cities, bacteria in these biofilm communities talk with each other. They secrete small chemical molecules into the environment to communicate with each other and alter the behavior of their neighboring cells. And my lab interprets and harnesses this chemical language of bacteria. In many cases, these communication signals are only made when other bacteria are nearby. And so in the lab, we often grow more than one kind of bacteria together, trying to mimic the natural communities that bacteria live in. We want to exploit these chemical signals that bacteria are already using to communicate with each other, so that we can manipulate their behavior, prevent disease, and reduce antibiotic use. We want to know which chemical signals help biofilms form, which signals prevent biofilms from forming in the first place, or can break them down once they are already formed. If we can identify these chemical signals that eliminate biofilms, or that prevent other harmful pathogenic phenotypes associated with different diseases, we could use these compounds as drugs to improve human health. And just like we want to get rid of the poison ivy in our yard without using that flamethrower, we anticipate that the discoveries we are making in these kinds of studies will help us keep people healthy without the risk of developing antibiotic resistance.